Vitamin B9, or folate, is widely recognized for its role in preventing neural tube defects and supporting red blood cell formation, yet its deeper biochemical and epigenetic functions are often overlooked. One of the most intriguing aspects of folate metabolism is its central role in the process of DNA methylation. This process, which adds methyl groups to DNA without changing the underlying genetic code, acts like a switchboard for gene activity, turning certain genes on or off depending on cellular needs, developmental stages, or environmental influences. Folate is a major supplier of methyl groups through its participation in the 1-carbon cycle, particularly via its conversion to 5-methyl tetrahydrofolate. This methyl donor feeds into the production of S-adenosylmethionine, SAM, the universal methyl donor required for methylating not only DNA, but also proteins, neurotransmitters, and lipids. The implications of folate's role in DNA methylation extend far beyond basic cell function. Disrupted methylation patterns, either too much or too little, are increasingly associated with the development of cancer, cardiovascular disease, autoimmune disorders, and neurodevelopmental conditions. Aberrant methylation can silence tumor suppressor genes or activate oncogenes, making folate intake and metabolism relevant in cancer prevention research. However, what is less commonly appreciated is that folate's role in methylation also affects how we age and how resilient our gene expression is to environmental stressors. Through epigenetic regulation, folate shapes how our genes respond to diet, toxins, and inflammation across the lifespan. Some studies suggest that optimizing folate status might delay age-associated epigenetic drift and reduce the risk of chronic disease emergence later in life. Another lesser-known but compelling domain of folate activity lies in its effects on mood and mental health, mediated again through methylation. Folate-dependent methylation affects the synthesis of key neurotransmitters like serotonin, dopamine, and norepinephrine. These neurotransmitters require methylation for their biosynthesis and metabolism, and when folate levels are low, the efficiency of this system declines. This can contribute to symptoms of depression and cognitive decline, especially in genetically susceptible individuals. Interestingly, individuals with polymorphisms in the MTHFR gene, particularly the common C677T variant, have a reduced ability to convert folate into its active methylated form, impairing methylation-related pathways. This genetic detail turns the spotlight on folate not just as a general nutrient, but as a precision tool in personalized medicine. For such individuals, supplementing with L-methylfolate rather than folic acid may bypass the enzymatic bottleneck and restore normal methylation dynamics. Folate also plays a somewhat hidden but powerful role in the immune system. During immune activation, DNA synthesis and methylation ramp up as T cells proliferate and differentiate. Folate availability becomes rate-limiting during these stages, meaning inadequate levels can directly suppress immune efficiency. What's especially interesting is that folate doesn't just fuel the immune response, it helps shape it. Emerging studies indicate that methylation patterns influenced by folate determine how immune cells are programmed to react to pathogens, tolerate commensal microbes, or respond to inflammatory signals. Thus, folate is involved in immune education, subtly guiding how the body distinguishes friend from foe. This epigenetic training may have long-term effects on immune tolerance, allergy risk, and autoimmune tendencies. Even reproductive health contains an epigenetic layer where folate acts as a silent architect. In both men and women, folate status influences gamete quality, not just through chromosomal integrity, but also through methylation of DNA that gets passed on to the next generation. In men, low folate levels are linked to abnormal sperm DNA methylation patterns, potentially affecting fertility and offspring health. In women, adequate folate is essential not only for fetal development, but also for epigenetic imprinting during oocyte maturation. These findings suggest that folate is not simply a maternal nutrient, but a biparental epigenetic regulator, shaping how traits and risks are transmitted across generations. Finally, in the context of chronic disease prevention, folate emerges as a dietary compound with a strategic role in gene-environment interaction. Nutrition is a major modifiable factor in epigenetic aging, and folate, due to its methylation function, has been investigated as a dietary lever for modifying disease risk trajectories. In populations exposed to heavy metals or air pollution, higher folate intake has been associated with more favorable DNA methylation patterns and lower oxidative stress. This suggests folate might act as a buffer against environmental insults, adding an important dimension to public health strategies aimed at vulnerable groups. In all these areas, mental health, immunity, reproduction, environmental resilience, folate's value extends well beyond the prevention of anemia or birth defects. It is an architect of biochemical signaling, a regulator of gene expression, and a protector of generational health. Its seemingly quiet role in methylation belies a sweeping influence over how our genes function, interact, and adapt throughout life.